Good afternoon, everyone. Good morning, David. So it is our great pleasure to welcome you today. So David is an American science, nature, and travel writer, as well as the author of 15 books. He also collaborated for many years with several popular magazines and periodicals, including Outside, National Geographic, Harper's, Rolling Stone, the New York Times Book Review, The New Yorker. One of his most popular books, Spillover, published in 2012, describes the mechanisms of zoonosis, a natural process by which some animal pathogens evolve and acquire the capability to infect and transmit to the human species. This book became extremely popular earlier this year after the start of COVID-19 pandemic, since it predicted the possibility of a new global pandemic originating from coronavirus. We will get back to Spillover later in the discussion. However, today we will mainly discuss with David about his most recent book, The Tangled Tree, where he carries the reader along an amazing journey throughout the history of life, from the foundation of evolutionary biology by Charles Darwin to the most recent discovery of contemporary biology that are projecting the Darwinian paradigm into a radically new perspective. So, David, in your book, you discuss the tree of life. What was your motivation and driving force to write this book?
Well, thank you, Francesco, and thank you for welcoming me. Antonino, thank you for being with us this morning. I'm very, very pleased to be part of this conversation. The motivation for doing this book about the tree of life and the radical redrawing of the tree of life began when I first read about a very strange phenomenon called horizontal gene transfer. I had finished my book on emerging diseases, pandemic viruses, Spillover, as you mentioned, and it was published in 2012 in the US. And then I was looking around for another book project. And in early 2013, I read some articles about this phenomenon, horizontal gene transfer. What? I thought, genes moving sideways across species boundaries, genes moving sideways even from one kingdom of life to another. And I thought immediately that's impossible. I know, I've studied enough evolutionary biology. I know 3 reasons why that's impossible. I read a little bit more and I saw that indeed it is possible. Genes do move, and over the course of evolutionary history have moved sideways across boundaries between species, even between kingdoms. How is that possible? Well, we can get into that. But first of all, it was this discovery of this phenomenon that had such implications for evolutionary history and the shape of the tree of life, the evolutionary picture of life as a tree. So I started reading more about it. It was fascinating, and I thought maybe I can do a book about this, but it was so counterintuitive and surprising and somewhat complicated that I thought, now how can I do a book? And then I discovered a character, a human person, Carl Woese, a molecular biologist, really a microbiologist, first of all, in Urbana, Illinois, at the University of Illinois, a little man that nobody outside of science had ever heard of. And I started reading about his story in some technical works. And he became, and he, I realized, was the most important biologist of the 20th century that you've never heard of, and that I had never heard of. Extraordinarily important and connected to this phenomenon of horizontal gene transfer and redrawing the tree of life. So then I realized I can write about this by telling the story of Carl Woese and the people he worked with and the discoveries that they made that led to this whole new understanding that does not overturn Darwin, but expands Darwinian understanding of evolution in a huge new way.
David, listen, since you said twice, redrawing the horizontal gene transfer redraws the tree of life and expands the tree of life. So maybe we can start from what was the intuition of Charles Darwin when he wrote in his notebook his, I think, quotation mark, and he drew this beautiful sketch. So
Right.
that is where our conception of tree of life starts. Maybe we can start from there and then say why horizontal gene transfer breaks this shape.
Perfect. Yes, good idea. So Charles Darwin came home from the voyage of the Beagle in 1836, this 5-year round-the-world voyage that had taken this young, very conventional Englishman who was interested in natural history and geology, and showed him things in the natural world, including the species on the Galapagos Islands, that changed his old-fashioned religious conception of the origins of species, the origin of diversity on Earth, the idea that God had created all these creatures individually. Suddenly he was challenged to think in a new way and to say, no, I think that species evolved, diversity, adaptation evolved by natural processes. He did not yet have a theory how that could be done, a mechanism for it. And so back in London for 2 years, living in a house that essentially his father was paying for. He didn't have a job. He was impassioned to think about this. And he started a series of notebooks, little notebooks, just little pocket notebooks that became known as his transmutation notebooks. He was writing about his reading, writing about his thoughts. How can this be? How can diversity have arisen naturally? And in 1838, as I recall, in his B notebook, the second one, he drew a scrawled picture of a tree, just a very simple picture, like that, in his notebook. And beneath it, he wrote, "I think." And what he was saying was, I think that all creatures, all biological diversity, have arisen from common origins by a process of divergence, branching, splitting. Lineages have gone through time and reached points where a single lineage of creatures has split into 2 different kinds of lineages. We call that speciation now. And that's how diversity has arisen. And then around the same time, he got the idea of evolution by natural selection. How could this change happen? How could this branching divergence happen? And he realized that it's a matter of there is variation within populations of individuals. He wouldn't say genetic variation, but moths of one species have tiny little differences, one from another within that species. And then there's competition for resources. So the moths in that species are competing with one another. And there aren't enough resources for all of the caterpillars born to those moths. So they're competing. And that competition leads to the survival of the fittest, that famous term, so that the individual differences determine which moths survive, and that creates the possibility of divergence. Some moths are better living on this kind of a tree. Some moths are better living on this kind of a tree. Suddenly you get separations, and then these lineages become more and more different over time. And that's the divergence. So that is the classic Darwinian evolutionary tree of life. It is all about divergence, divergence, divergence. This is my very, very sophisticated audiovisual aids. This is my PowerPoint.
Best one. So you told us very, very, very well how the tree organization, vertical transmission of genes as opposed to horizontal, how horizontal gene transfer, you explained us previously how this in a way expands and allow forces us to redraw but before coming again back to horizontal gene transfer, the tree-like structure of life was for over a century based on morphological aspects. Then at some point, something very important happens, and here is where your Carl Woese, and so that's where molecular phylogenetics comes in. So maybe this is a second validation, a very strong validation of the tree structure, but it also allows more structure to be gained and more information.
Right.
So how is this coming?
Exactly, exactly. Well, what happened was that, as you said, Antonino, through most of the first half or 2/3 of the 20th century, evolutionary biology was done on a morphological basis, mostly. Creatures had different shapes, different characteristics, different physical aspects that would distinguish one species from another. And through the fossil record of the shapes that had existed in creatures in the past, we could see the history of change. That was the primary evidence, not the only evidence, but the primary evidence for deciding the shape of the tree of life. And then came the rise of molecular biology. Watson and Crick in 1953 solved the structure of DNA. Other scientists, Francois Jacob, Jacques Monod at the Pasteur Institute in Paris, made very important contributions. And so there was the rise of molecular biology, the idea of understanding biology at the level of molecules, proteins, and nucleic acids such as DNA and the other form of nucleic acid, RNA. These two molecules that are information carrying molecules. Proteins, of course, are long, complicated molecules made up of many different components. DNA and RNA are also long, complicated molecules made up of just 4 different components arranged in different orders, infinite possibilities. And it was understood that those are the information carrying molecules. And then a couple of scientists before Carl Woese gave hints to the possibilities of using those molecules. In 1957, Francis Crick of Watson and Crick wrote a paper about protein synthesis. How are proteins created in cells? And he mentioned in passing that these long, complicated molecules, proteins, might carry some information about the history of life and about the relatedness of life, one kind of creature to another. And he called that protein taxonomy, the idea of organizing, classifying creatures based on the proteins found in them. And then Linus Pauling, who won the Nobel Prize twice, I believe, and a younger colleague of his named Emil Zuckerkandl, in the early 60s, advanced the idea of chemical paleogenetics, using molecules, using chemicals to understand the ancient genetics of creatures. And then along came Carl Woese, influenced by them, and he got the idea of doing what he called using the internal fossil record of a cell to determine the earliest history of life. And he mentioned that in a letter in 1969 to Francis Crick. Francis, he barely knew Crick, but he corresponded with him, and he said, Francis, I have this idea of pushing the history of life back 1 billion years earlier than what we can understand by using the internal fossil record of a cell. And again, he was talking about using complicated molecules to discern which living creatures are most closely related to which others, and what their lineages might look like going back into the past. And then he made one very, very brilliant choice. He chose the right molecule. There is a little organ, a number of little organs sprinkled like pepper in all cells of cellular creatures called ribosomes. And ribosomes are the little machines, I call them the 3D printers, that take genetic information and they turn it into physical reality. They take RNA that has been copied from DNA, and the RNA runs into them like tape instructions, and out of the ribosome comes a protein, a 3-dimensional physical reality, turning information into physical reality. What's important about ribosomes is that every living cell on Earth has to have them in order to turn its genetic code into its body, its physical components. So Carl Woese said, if all living cells have ribosomes, and if ribosomes include some structural components made up of RNA, not just for information, but for structure in the ribosome, RNA, this information molecule, then I could extract that RNA from ribosomes of different creatures, and I could find a record of the deepest history of life. And so that's what he started to do in his little laboratory in Urbana, Illinois, using all sorts of primitive, dangerous methods to sequence, to extract this molecule from this little organ, the universal organ, ribosomes, and then having extracted that, to sequence at least fragments of it, and then make collections, paragraphs of the fragments, and compare one to the other. And that's what he started doing in the mid-70s, and that essentially was the birth of molecular phylogenetics.
So, David, as you already said, Carl Woese used molecular phylogenetics as a sort of quantitative tool to trace back to the origin of life. So this was his overarching goal of his whole research. In this process, he made an outstanding discovery that started to really expand the tree of life in an unexpected way. So how did the tree of life look like after Carl Woese?
There's the old tree of life. Two great kingdoms of life, bacteria and everything else. Two great limbs diverging from the common trunk. And that was what was thought of the shape of the tree. The most gross approximation of the tree of life is that there was a trunk and there were two great limbs. Bacteria, which are cellular creatures that do not have complex internal anatomy, and then what we call eukaryotes, which is animals, plants, fungi, and everything else that are cellular creatures. They can be multicellular or they can be single cellular, but they have complex internal anatomy. They have cell nucleus. They have mitochondria. If they're plants, they have chloroplasts, these complicated things. They have these other internal organs, just as we have kidney, liver, spleen. They have internal organs. Bacteria, it's just a bag of of molecules. But that's not fair to bacteria, but that's roughly true. So Carl Woese came along and he said, okay, now I'm going to use my method to discern the relatedness of different creatures. And I'm going to grow some creatures in my lab. I'm going to extract their ribosomal RNA. I'm going to sequence it, and I'm going to see what's closely related to what else. So he started doing that, and he sequenced a number of bacteria, and he could see which bacteria were closely related to which others. And then there were some other bacteria that were very peculiar because they metabolized methane. They had a sort of a sulfur economy. They lived in strange places like hot pots in Yellowstone Park, hot springs. And so with help from some colleagues, he grew some of those in his lab, which was very difficult. And he sequenced the ribosomal RNA, and he put it up on a screen. These different fragments of the ribosomal RNA he would print them out on a sheet of X-ray film, and they would make a pattern that looked like a galloping herd of amoebae going down a racetrack. Some ahead, some behind, some over here, some over there. And there were these ovals that indicated the different fragments. And he looked at the X-ray film from this strange bacterium, and it was all wrong. It did not have the right ovals in the right positions. It had a completely different sort of signature, of genetic signature. And he immediately said, he walked down the corridor to his colleague, Ralph Wolf, and he said, "Wolf, these things aren't even bacteria. For 300 years, these creatures, seen through microscopes, had appeared to be bacteria. Carl Woese looked at their genetics, and he found that they were more closely related to us, to human beings, than to bacteria, although through a microscope they look like bacteria. So he realized that there was a third kingdom, that the kingdom of life does not look like this, or the tree of life, but the tree of life looks like this. There's a third major limb, and that became known as the archaea, because at first these were thought to be the most ancient kinds of creatures. So in 1977, he published a paper with his postdoc, George Fox, who was one of my sources for my book. Carl Woese was dead before I began the book, but I spent time with his colleagues and students. George Fox was his colleague for this discovery. They published in 1977 a paper announcing that using their method, they had discerned that there were not two great kingdoms of life, bacteria and eukaryotes, but three, including the archaea, which were completely distinct from the other two. And people all around the world, especially in America, said, Woese, you're crazy, you're an idiot, you're wrong. Scientists in Germany, who had been studying these methane creatures and noticed that they were peculiar, said, no, we think Woese is right. And eventually, as more scientists worked on this, this discovery was proven to be correct, that there was a third great domain or kingdom of life, the archaea, and he became very famous. He was on the front page of the New York Times, and that was the peak of his fame in November of 1977. There was a picture of him on the front page of the New York Times with his feet up on his desk, in tennis shoes in his office, and it was for this discovery. It wasn't the most important discovery that he ever made, but it was the most famous discovery he ever made.
So here comes a bit of a paradox, because you have a scientist at the peak of his fame, as you say, that is on headlines of journals, that he has described these three domains of new domains of life. And then the new evidence on horizontal gene transfer starts creeping in. And this shakes the very structure of the tree, because if horizontal gene transfer occurs, how would this change the structure of the tree? How would it change the real relationships of organisms one to the other? What are the implications? How frequent it is? And also, okay, and how was Carl Woese's reaction to this?
Yes, very, very good, interesting question. First of all, this method that Woese had created, extracting ribosomal RNA, sequencing it in fragments, comparing the fragments, using genome sequencing generally as a tool of phylogeny, which is the drawing of trees of life. So this became molecular phylogenetics. It caught on and it spread, and people repeated his methods and improved his methods. And genome sequencing improved. It became less primitive, less slow, less dangerous. Computers, personal computers arose with great calculating power. So this method became more and more capable of looking at all different kinds of creatures, comparing their genomes, and drawing the lineages, drawing the tree. And one of the things that scientists noticed was that occasionally there were genes out on a little branch that shouldn't be there because they had not been present lower on the limb. They just appeared. And their predecessors or their other ancestors, their closely related ancestors, didn't have this gene, but suddenly they had this gene. And so scientists ask, where did that gene come from? How did it get into that creature if all of the creature's cousins, living cousins, don't have that gene? How did this one creature suddenly get it? Where did it come from? And the answer was horizontal gene transfer. It had come in sideways. And as you mentioned, Antonino, the the old-fashioned Darwinian idea of inheritance was vertical in this sense, limbs rising up and branching, diverging. Horizontal meant flow of genes sideways from one limb to another. So scientists recognized that this was happening and had been happening probably throughout the history of evolution. Here comes some more of my very fancy high-tech graphics. So now we were saying instead of this, we had also this. Genes moving sideways from one kingdom of life, from one species into another. How was that possible? Well, the great molecular biologist Joshua Lederberg and his wife, Esther Lederberg, had coined a term earlier on when they were studying the genetics of bacteria, and they called it infective heredity. Infective heredity. So the simplest explanation, there are complicated explanations, but the simplest explanation for much of this horizontal gene transfer is that it had involved infection with viruses or bacteria that sometimes carried genes with them from one creature to another and deposited those in the second form of creature they infected, or the lineage of viruses infected, or deposited some of their own genes in a creature that they infected. That is one of the major forms of horizontal gene transfer. And so this exploded, and more scientists started doing this method, sequencing, finding more and more evidence of horizontal gene transfer. Meanwhile, Carl Woese didn't like this quite so much because it blurred the picture of his great discovery of the archaea. Suddenly, well, all these things are kind of connected, and the tree of life is tangled, hence my title. And so toward the end of his life, Woese started to put he had been putting his foot on the accelerator, on the gas pedal, in terms of discoveries that were possible through molecular phylogenetics. And toward the end, it seems that he was starting to put his foot on the brake a little bit and saying, wait a minute. Yes, horizontal gene transfer was very important at the beginning of life, right at the beginning. Maybe not so important later on. And that continues to be a very lively, forceful discussion. There are still brilliant scientists who say horizontal gene transfer was important at the beginning of life. It's important among bacteria and archaea. It's not very important among complex creatures. And others say, no, look at the evidence we have. We have a lot of evidence that has been very important in complex creatures as well. It has been one of the phenomena that have been extremely important in terms of major transitions in evolutionary history. And you can't understand evolutionary history unless you take into account horizontal gene transfer and the entanglement of the tree of life.
Thank you. This is, you are throwing a very big stone, because you cannot understand evolution if you don't take into consideration horizontal gene transfer. It's a very important statement, because evolution is what brought us here. And we look forward and we look backwards. So you mentioned two aspects in which horizontal gene transfer, the scientific community is discussing it. One is the early evolution, and the second aspect is complex creatures. Let's take the two aspects one by one. Let's look backwards. So looking at a tree, even if it is a tangled tree, a tree has a root. And so the structure of the tree brings us naturally to ask the question, how is the root of this tree? So we call this LUCA, the last universal common ancestor. How can we use the tree of life to trace back the properties of our last common ancestor? And the complementary question is, how would horizontal gene transfer in a very, very early phase of prebiotic or early biotic evolution play a role? Because we all think that the origin of life is a natural physico-chemical process that is part of the geological, geochemical evolution of our planet. So the two things must go together.
Yes. Okay, well, I will try and answer. But I should also remind our audience that you two gentlemen know molecular biology better than I do. This is an enormously satisfying interview for me to have, because you two know this subject so well. You know, and you can probably answer your own question better than I can, Antonino.
No, I cannot. I'm a neuroscientist. So anyway.
First of all, what you're talking about, we go back 3.5, 3.8 billion years. I think now the earliest evidence we have of cellular forms of life come from about 3.8 billion years ago, a very long time ago. What do we know about that? Well, mostly what we know is by very intelligent, brilliant inference from, not from the fossil record, because you can't tell very much from 3.8 billion year old rock that has what seem to be cellular forms in it. All you can really gather from that is that maybe there were cellular forms of life 3.8 billion years ago. But what did they look like? How did they evolve? Where had they come from? There are people like Bill Martin, William Martin in Dusseldorf, and a number of others who have proposed different theories about how cellular life began. Did it begin? It needed chemical components, chemical complexity, it needed energy, it needed perhaps some sort of a physical structure that would be transformed into cellular structures. Bill Martin and his colleagues have hypothesized that possibly this began at deep thermal vents in the oceans, where there was sulfur, where there was heat, where there was tiny cellular-like spaces in the rock around the vents. We don't know. That's an ongoing conversation. But Carl Woese and a number of others, Walter Gilbert, other scientists around the world, they embraced an idea that they called the RNA world, the RNA world. So they hypothesized that before cellular creatures, before creatures that were encased in cell walls, evolving by natural selection, there was a stage when complex molecules started to assemble and to replicate themselves. They speculate that the earliest complex molecule doing this was RNA, this single-stranded relative of DNA. RNA is a nucleic acid that's composed of 4 letters, 4 nucleotide bases that we give letters to abbreviate. And just as DNA is A, C, T, and G, RNA is composed of letters A, C, U, and G, and they stand for these nucleotide bases. So you've got A, C, U, and G, A, C, U, and G, repeating in different combinations. The hypothesis of RNA world is that somewhere, maybe at the bottom of the ocean or in warm ponds or wherever, there was a richness of molecular components and energy, and RNA molecules started to self-assemble, putting together these components. And then when they had self-assembled, they could replicate themselves because of the way that, these nucleotide bases fit together. They could make copies of themselves. And they started pulling in these different molecular components. One string of RNA would pull in some, and another string of RNA would pull in some, and there was competition. So there was natural selection. There was variation, difference from one string to another. There was competition for resources. There was a finiteness of resources. And so one individual, not a cell, not a creature, but an individual string of RNA might have succeeded better than another. And the RNA world encompasses that idea. It's hard to prove. It's a hypothesis, but it's an important hypothesis. It's not the only hypothesis. And from that then came eventually DNA and cellular creatures and the earliest cellular forms of the tree of life. And it's possible that there were different kinds of cellular creatures composed perhaps with different genetic codes, and that they were competing against one another, and that one particular lineage of cellular creatures that used DNA and used the code ACTG and created amino acids to put together proteins using particular combinations of those letters, that particular lineage of cellular creatures competed successfully against any other form of cellular creature and drove them to extinction, and then became the root of the tree of life that we know, became LUCA, the last universal common ancestor. And from that, all the rest is sprung. This is hypothetical, but this is the scenario that that these brilliant scientists have proposed.
Yes, because otherwise, if LUCA was just one system that was the winning one, you cannot think how by chance it would have been the only one. So of course what you say is very reasonable to think that there might have been many different systems, physical-chemical systems going on in prebiotic life.
Yes.
And then a winning system in a way was a winning combination that competed with the others. But let's now look present and forward. Maybe Francesco, you want to ask something about looking at now and looking forward instead of looking backwards in the tree of life.
Yeah. In this period, I mean, when we can witness. We live in a historical moment where biodiversity is particularly challenged by, is particularly impacted by human society, which is destroying probably in a irreversible way many ecosystems. On the other hand, synthetic biology holds the promise to create even more biological diversity than the one that has been sampled through natural evolutionary trajectories. So if we now want to look forward into the future, can we use the tangled tree of life to sort of predict where are we heading, or at least use it as a sort of reference from where we stand now?
Yes.
To move on.
Francesco, I think we can certainly use it as a form of reference. Whether we can predict from it, I don't know. Let's talk about that a little bit. First of all, before I wrote this book, before I researched and wrote this book, I was deeply concerned about biological diversity, the biological diversity that we have on this planet. I was skeptical of synthetic life, of the technologies of genetic engineering, recombinant DNA that move genes from a jellyfish into a tomato in order to make it more frost resistant or whatever. I was very skeptical about that, uneasy about that. And then when I discovered that horizontal gene transfer is a natural phenomenon that's been going on for, 3.8 billion years or so, it sort of dissolved my resistance to synthetic biology, at least on the grounds that it's unnatural. It's not unnatural. It's been happening. It's just that now humans are doing it rather than it being, rather than viruses doing it, viruses causing it or whatever. So my mind has opened more to synthetic biology. I realize it can't be criticized and condemned on the grounds that it's unnatural. There has always been this mixing. There's nothing sacred about the genes in a particular lineage or the genes in a particular species, if you believe in Darwinian and Wozian evolution. There's nothing sacred because genes have been moving sideways from one lineage into another. So if humans do that, you might say that it's unwise because it's hard to predict the consequences, but you can't say that it's unnatural. That's the way that this body of work that I describe in the book is a framework for thinking about what's going on now. In terms of synthetic biology, adding biological diversity to compensate for the catastrophic, tragic loss of biological diversity that humans are causing, my prediction is not optimistic about that. I think that there are amazing things that will be done with synthetic biology. Some of them will be very beneficial to humans and possibly beneficial to ecosystems too. Engineering new ways for bacteria to eat spilled petroleum. Engineering new ways perhaps for bacteria or other microbes to clean up radioactive waste. Engineering other things that are not just good for humans, but good for biological diversity. So those things are possible. But in terms of adding biological diversity on the scale that we're losing it, and adding biological diversity that has one of the most important characteristics that I consider valuable and many other people do, and that is the sheer wonder, the sheer arbitrariness, the sheer unexpectedness, the sheer gratuitousness of biological diversity. Things that are beautiful and amazing because they exist and because they are different from everything else, things that no human could ever have imagined creating, but things that time and evolution have created. I don't think that humanity, for all the brilliance of the science that we have now, will be capable of producing those kinds of unexpected wonders in the diversity and abundance of the things that we're losing.
Thank you, David. You mentioned horizontal gene transfer as mediated by viruses in general, being or mobile elements, or you need biology has used technology, biological technology, to move pieces of DNA between cells and between organisms. And this is exactly the job that viruses do. So can you think of viruses in a broad sense, replicable nucleic acid elements, as natural actors of evolution? And if you see them in this way, then they are the virosphere is natural participant of our biosphere and should only be seen as a threat when it affects our health, of course, as it does. But in a way, we should look at them also as actors that shape the evolution. For instance, can you maybe elaborate on the fact that 8% of our genome contains pieces of DNA, genes, or sequence elements that are of retroviral origin? So 8% is a lot. So maybe you can comment and elaborate on that and tell us your views on that.
Absolutely, Antonino. I'm exploding with energy to answer that question, and I'll take it in two parts. The part about humans, the role that viruses have played in human evolution, that 8% of our genomes are retroviral DNA. Let me take that second. First of all, you're talking about the virosphere and the important roles generally that viruses have played. And that's absolutely true and important. And in fact, by coincidence, I've just finished spending a couple of weeks, probably a month, writing writing an essay for National Geographic on the subject of the evolutionary origins of viruses and the role, including the beneficial role that viruses have played in the history of evolution. And it gave me an opportunity to talk to some very, very interesting thinkers on the evolutionary origins of viruses. Jean-Michel Claverie in Marseille, and Patrick Forterre at the Pasteur Institute in Paris, and some others. Their work and the work of their colleagues shows us that, first of all, viruses are the greatest trove of genetic diversity on the planet. Viruses are sources of genetic innovation. Viruses are one of the crucial places, if not the most crucial place, where new genes, new arrangements of DNA and RNA come from. Where do viruses come from, and where do they fit on the tree of life? Again, that's another really complicated story. Back when we talked about different cellular lineages at the very beginning that competed against one another, and one of them won, perhaps, and became the last universal common ancestor. Well, there are some who think that possibly there was another cellular lineage that lost that competition, and they survived in just reduced form by becoming viruses, by becoming replicable genetic elements that couldn't replicate on their own, but could only replicate as parasites of cellular creatures. Another interesting idea. So viruses are hugely important. Horizontal gene transfer, we've talked about that, and I talked about infective heredity. That is one of the main, but not the only way that horizontal gene transfer happens. Three technical terms for the different methods: conjugation, transformation, and transduction. Conjugation, transformation, conjugation means that essentially that bacteria or archaea are trading genes sideways, almost as though they were mating, but mating across species boundaries, sending little packets of genes sideways, literally through a tube that's like a penis. That's conjugation. Transformation is when a cell busts open and its DNA is floating around loose in some sort of an environment, and maybe some other creature, a simple creature, absorbs that DNA and puts it to use. That's transformation. And transduction means a virus takes a piece of DNA from one creature and carries it and replicates it as it replicates, and then eventually deposits it in another creature. Transduction by viruses, moving DNA from one creature to another. Retroviruses are, as you know, viruses that move backward, retro, by inserting they have genomes of RNA, they convert the RNA to DNA, and they insert that into the genome of the cell that they have infected. All viruses replicate by being parasites on cells, but generally they don't insert their DNA into the genome. A retrovirus inserts its DNA into the genome of the cell that it infects. If it's infecting immune cells, then the immune cells replicate that DNA, and eventually the person, if the retrovirus is HIV, then the person begins to suffer the symptoms of AIDS. HIV is a retrovirus that infects immune cells. But over the course of animal evolution, there have been retroviruses that have infected reproductive cells, what we call the germ cells of creatures, the egg cells, the sperm cells, the stem cells that create egg and sperm. And when a retrovirus inserts its DNA into that, it becomes heritable. It can be passed on to offspring. And that has happened enough in the course of animal evolution that we humans now contain 8%. 8% of our genomes are genes that have been inserted into our genome by retroviruses. So now we're back to that 8%. Some of those genes are silent. They don't seem to have a function, at least that we know of. And some of them are very important and have been adapted for purposes in animals, in mammals, in primates, in humans, including a gene called syncytin 2, syncytin, syncytin 2, that was originally an envelope gene for a retrovirus, so it created an envelope for the viral particle. It infected animals. Different versions of it were replaced, added, improved as animals turned in, some animals turned into mammals, and some mammals turned into primates, and some primates turned into humans. And now we have this improved version called syncytin 2, and instead of creating a viral envelope, it creates a membrane around the placenta in humans, without which pregnancy cannot be successful, without which pregnancy is impossible. This syncytin 2 creates a membrane that helps to mediate between the fetus and the mother. It transports nutrients in across the placenta to the fetus. It transports waste products out so that the mother can excrete them. It also probably protects the fetus from the mother's immune system. The fetus has a genome that's only 50% similar to the mother's, and so her immune system might react against it. This membrane from a viral gene protects the fetus from the mother's immune system, making possible mammalian life, human life.
There is you mentioned, and you described this very well in the book, and you describe it now, syncytin as a retrovirally originated or linked gene. It reminds me, in the nervous system, there is a gene that is called ARC, and this is a very important gene involved in synaptic plasticity and in memory. And very recently, people have discovered that this protein that works at synapses assembles into viral-like particles, and the sequence of ARC has domains that are very, very similar to the GAG domain of the HIV virus. So now this is a very hot field in neurobiology to try to understand these viral particles, if they shuffle around between neuron cells, RNAs from cell to cell. So this whole technology of viral transfer of genetic material could also occur within an individual, which brings me to another question, if I cannot overtake my time, but which is a more philosophical one. So this phenomenon of horizontal gene transfer, regardless of how relevant in relative terms it is with respect to the classical vertical gene transfer, however, it definitely challenges our notion of what a species is, what an individual is. So an individual is something that is open, according to this view, is open to genetic exchanges, and so this also reminds us of the microbiome. We all are organisms that have a host of zillions of bacteria that live with us and evolve with us. So what's your view of how to define today a species, an individual, and within the context of the Tangled Tree?
Yeah, well, absolutely. Those are questions that this whole field of science raises. And I say at the end of my book, there are three categoricals, three things that have been considered absolute truths or absolute realities. One is a species. A species is a population of creatures that can't mate productively with other populations of creatures. They are discrete, they are separate. Black line, solid black line drawn around them. An individual is also supposedly a categorical. You're an individual, I am an individual, Francesco is an individual. We have boundaries that are inviolate. And the third categorical is that the history of life is shaped like a tree. And the work of Carl Woese and his colleagues and the people that he influenced and the people that have come after him, all these scientists, all of this work that I describe in the book has shown that all three of those categoricals are non-categorical. The boundaries are not solid black lines. There is
so would you
truth.
say they get fuzzier?
They get fuzzier. Yes, absolutely. Those lines around the individual, the lines around species, the tree gets more tangled, and the individual turns out to be a community of creatures, and the species turns out to be something that does have genes coming in from sideways, from other lineages, not just from individuals of the same species. And inheritance is horizontal as well as vertical. So as you said, we now know that we humans, we have a microbiome. We've got thousands of different bacteria and probably some archaea living in our bodies, living on our skin, living in our eyes, living in our nose, certainly living in our stomach and in our intestines. We've got all these bacteria and other creatures, fungi, that are natural, healthy parts of the ecosystem that is us. And when we get sick with a bacterial infection, it's not because there is one bacteria that is in us that shouldn't, and bacteria shouldn't be in us. It's because there's a particular bacteria that has exceeded its balance, that is causing disruption in our ecosystem. We need to reestablish that balance. And of course, not just in our bellies, not just on our skin, not just in our mouths, but in our genome, we are also mosaics. We are composed of a multitude of other creatures that have come together over the course of 3.8 billion years to create cells from which our cells are descended. Even our mitochondria, the little organs within our cells that package energy, we know those are captured bacteria. That's a part of this story that we haven't even talked about, but the theory of endosymbiosis, the idea that internal organs of complex cells, the mitochondria in all creatures, all cellular creatures, and the chloroplasts in plants, those are captured bacteria that have become part of our cells over the course of evolutionary time and adapted to become symbiotic parts of us. So we are many.
So let me now bring back your spillover book, David, and talk about what is happening in this period, in this month. So the global pandemic.
Yeah.
So in spillover, you mentioned the next big one. So talking about the new emerging viral diseases, jumping from animal species to the human. So what do you think are the breeding grounds for global pandemics? Is it the current pandemic, so the big one you were mentioning in spillover, or shall we expect something bigger? And also, trying to link with this horizontal gene transfer topic, shall we consider the possibility that the current pandemic is already shaping or is going to shape our evolutionary trajectory through, horizontal gene transfer or other genomic integration or genetic mechanisms by the SARS-CoV-2 virus?
Okay, well, I hear three questions there, Francesco, and let me take them in order. First of all, what are the conditions that are causing pandemics, including our current pandemic? The spillover of dangerous viruses from non-human animals into humans. This is a category of viruses we call zoonotic viruses. That means animal infections that are transmissible to humans. 70% of human infectious diseases fall into this category. They are infections that we get essentially from animals, mostly wild animals, but in some cases domestic animals. And they spill over into humans, and then if they discover they can replicate within us, might be a bacteria, might be a fungus, but in the most dramatic cases, it's a virus. If it can replicate in us, it can pass from one individual to another, then it becomes a human disease. It becomes an outbreak that might lead to an epidemic that might lead to a pandemic, as we have now with COVID-19. The driving factors are human disruptive contact with wild animals. And that means invasion of diverse ecosystems, exploiting resources from the natural world, drawing more and more resources from the natural world to us. There are now 8 billion of us humans on the planet. We are very smart, we are very hungry, we're greedy for resources. So we're constantly drawing resources from the natural world. We're drawing meat in the form of wild animals. We're drawing timber, we're drawing minerals. We're drawing all of these things, fossil fuels, from the natural world. And as we draw those toward ourselves, we're also drawing viruses, new viruses toward ourselves, because all wild animals carry unique viruses, some of which can be very dangerous in humans, probably many of which can be very dangerous in humans. So by doing that, by drawing resources to ourselves, we give these viruses the opportunity to spill accidentally from their natural host into humans as a host. And that's what has happened now with the SARS-CoV-2 virus, the cause of COVID-19. When a virus gets into us now, because we live in dense aggregations in cities, and because we travel everywhere quickly, a virus can circle the world in 18 hours. And therefore, there is great danger of pandemics. And this one, I did mention that scientists that I spoke with and I used the term the next big one 10 years ago when I was researching spillover, the next big one. And the scientists that I talked to predicted to me several things about this next big global pandemic. They said, it'll be caused by a virus coming out of a wild animal. What kind of a wild animal? Well, very possibly a bat. What kind of a virus? Well, very possibly a coronavirus, because they evolve very quickly and they have a history of infecting humans. Where might that happen? Well, possibly at or near a wet market in China. All of this, they said to me, and I put it in the book 10 years ago, 8 years ago. And so now we have COVID-19, which is exactly what they were predicting to me, and I reported their predictions 8 years ago in spillover, COVID-19. So this is the next big one from the perspective of 8 years ago. Now, is it the last big one? No, there's no reason to believe it's the last big one. There are many, many more coronaviruses living in wild animals. We know that, many of which can also be dangerous to humans, different enough from this that once we get a vaccine for this virus, that doesn't mean it'll be good for the next coronavirus. We have to learn to work more quickly and more intelligently and with greater political will to protect us against these pandemic threats, developing generalized viral platforms or vaccine platforms so that we can adapt a generalized framework of a coronavirus vaccine to a new coronavirus when it appears. I don't say if it appears, I say when it appears. Diagnostic tests, we have to be much better in terms of producing diagnostic tests that are fast and accurate so they can be used immediately at airports and other places when an outbreak occurs. And we have to organize our national and our international responses to this much better. We have a lot of work to do to protect ourselves from the next of these pandemics. Now, the third part of your question, whether horizontal gene transfer needs to be part of our thinking. Now, it's possible that with all the people that are being infected by this virus, that horizontal gene transfer could occur, but the coronavirus does not, as far as we know, does not infect germ cells, does not infect reproductive cells, does not infect eggs or sperm or the stem cells that produce them. It targets very particular cells that have a very particular kind of protein on their surface, a particular kind of receptor that the virus can latch onto, so that they are infecting mostly our respiratory cells, and in some cases, some of our organs, apparently, kidneys and livers, possibly. I think that I don't want to make too much of a claim that my two most recent books are closely connected in terms of predicting the future. What I write about in Spillover is what's happening now. What I write about in The Tangled Tree is what has happened over 3.8 billion years. I'm not going to say that, oh, now we have to be worried about horizontal gene transfer since people all over the world are infected with this new virus. We've got a lot of things that are more urgent to worry about right now than that.
I think, David, that is a very, very important message. Horizontal gene transfer is a beautiful scientific concept that has shaped our evolution over the history of life. And the current outbreak is a different matter. And let me, since you mentioned it, let me also ask a question, as Francesco did, related to the current situation, because we cannot avoid this. So lessons learned, not from this one, but from the previous outbreak of SARS in 2003. By looking at the antibodies that have been developed against the spike protein of CoV-2 of the current virus, most of these also cross-react with the spike protein of of the SARS-CoV of 2003. And in fact, the first one that were published over April and May were so quick to be published because they were antibodies that were in the freezers of labs because they had been generated back in 2003. So we all know that the vaccine against SARS-CoV of 2003 and 4 was not developed because it was stopped in 2009 or 10, part because of non-industrial interest, but also part because of 3 or 4 million euros or dollars that the national agencies did not grant to pursue, simply because the epidemics had been stopped. So had those vaccines not been stopped, we would have a stock in the freezer. They might not have been optimal vaccines, but they would contain offer some protection. So do you think that this is really something, you mentioned it in your previous answer, you said we must develop a platform. So really, do you think that this was a sliding door that in 2003 humanity missed and could have avoided the current pandemics or softened the consequences of?
Yes, I do, exactly, probably so. It was tragically short-sighted. We know that generally vaccine efforts tend to be a big concern when there is an outbreak of a viral pathogen, and then if the outbreak is controlled and it goes away, then the incentive for the vaccine development tends to go away too. The same thing happened with Ebola. There were Ebola outbreaks occurring beginning in 1976 in remote parts of Central Africa. Why was there never a vaccine developed? Well, because vaccine makers thought, well, there's no money in developing a vaccine for a virus that kills 300 poor Africans 1 year and then disappears for 5 years into its reservoir host. So alas, tragically, not until 2014, when there was an epidemic in West Africa that terrified the world, was there real devotion of resources to an Ebola vaccine. There were some people who were quietly working on an Ebola vaccine, but they didn't have great support. So now we have an Ebola vaccine that is being used. Yes, absolutely. If we had had a vaccine developed for SARS-1, that would have been, I'm sure, enormously valuable in the situation of SARS-2. And we didn't have it because politics and business don't always listen to science.
Yes, that's That's an important point. At least we hope that the outcome of this tragic situation that we all hope we shall get through in the near future.
That's right.
Makes politics and science and pharmaceutical industry talk together. But of course, my own opinion. Now I'm saying an opinion, so that we can share. I think that the industry must be the industry, must think of the market. Governments must think of public interest, and the two interests are legitimate interests, but they must match together. We must find a way that we cannot leave drugs and vaccines only to market decisions. This, I think, I have a strong feeling for this. And
yes.
The vaccine case is a very obvious case.
Yes, yes, and those are legitimate frameworks of thought. With the political leaders, sometimes there is great short-sightedness because they think, well, should I invest in pandemic preparedness if the pandemic is not going to come between now and the next election?
And the next election, yes.
Yeah, but we also have people who come out of business, like Bill Gates, among others, who have vision. Bill and his wife Melinda Gates have put huge amounts of money before COVID-19 began. They were putting huge amounts of money into vaccine development efforts. There is an organization called, I believe, CEPI, C-E-P-I, Coalition for Emerging Pathogens Innovation, or something like that. And they were saying last year and in past years that there is a pandemic coming. I mean, it wasn't just me and a few other writers who were saying there's a pandemic coming. Bill Gates was saying that too. He saw this, and he was putting hundreds of millions of dollars into efforts to expand vaccine development and other forms of preparedness. So sometimes there is a productive crossover between even some of the world's most successful business people and the amount of foresight, the amount of vision that it takes to support science and preparedness.
But you know, one point is also that today an industry has not interest to develop a vaccine. Now in the pandemics, they are sure that the governments are going to buy because there are billions of patients worth. But in the early days of an outbreak, as you said, the idea is that there won't be a market because the outbreak is going to be controlled. So I think that we should also think of different incentives to how an industry is going to go into a, if the rewards for developing a vaccine or a drug would be on the impact of that drug once it is out and not on the market, that would be extremely different. But I think that this is going to carry us a bit too far away. And let me close by thanking you. And just I ask you a final question on changing on a less dramatic scenario. So your books are written as stories through people. Your books walk on the stories of characters, of peoples. This one was Carl Woese and all the people who collaborated with him. So this is one characteristic aspect of your books, and you have personally met a lot of people in the scientific community, a lot of characters, a lot of stories you heard about. So you have now a very, I think, a big view on the scientific community as a community and on individuals that make this community. So what would you, in short, what is your opinion of scientific as a community and of scientists as individuals?
That's a very interesting question, Antonino. And I guess, first of all, obviously I have huge admiration for the scientific enterprise and the people who pursue it, who advance it, who do it. brilliant, courageous, imaginative people. But that's not the end of it. What really draws me to them is that they are also complicated people, imperfect people. And I think that one of the things that makes science such a great story, such a great body of stories, is not just the brilliance and the innovation and the discovery. That's all important. But the fact that science is a human enterprise performed by humans. It's a process of seeking and establishing knowledge performed by humans. Football is performed by humans. Ballet is performed by humans. Grandmaster chess is performed by humans. Major League Baseball is performed by humans. There is passion, there is competition, there is jealousy, there is insecurity, there is ambition, as well as deep passion for seeking the truth. All of those things together. That's why I love science and scientists, is because of the human complexity. The fact that they're imperfect, they're struggling as individuals just as we all are.
Well, thank you very much, David, for your time with us. And I can only conclude by really inviting you to Pisa on the occasion of your next book. And we hope to see you in person in front of our students and to continue our discussion. And then we are very glad to have met you.
Thank you. I'm very glad to have met you too. I would love to come to Pisa when it's possible for me to do that. I love Italy. I haven't yet been to Pisa. I would love to do that. Antonino and Francesco and all of you there, the other Francesco, SNS, thank you so much for welcoming me. I really have enjoyed this.